This video will be kind of short. It's just on chapter 17, which the AP test doesn't put too much emphasis on about bacteria and viruses. The one thing that will be the most important is here, kind of towards the end of this presentation, when we talk about a case of HIV and about how bacteria increase genetic variation. So pay special attention to the last 2 or 3 slides here when we talk about those subjects. But I think viruses are really cool. They're really, really interesting. This picture right here is a virus that has gained a lot of media attention, rightly so, extremely deadly. This is the Ebola virus. And this is what it looks like under high contrast microscopy. And the things that we talk about viruses today, you can kind of use as a reference point, because the Ebola virus behaves in much the same way. So first we're going to talk about what is a virus. So viruses are very simple. As we talked about, eukaryotes are pretty complex. Prokaryotes are a lot simpler than them. Viruses are even more simplistic in structure and construction than prokaryotes. They're basically a sack of DNA that's coated in protective proteins. It's really all they are. And they have several important features that we'll look over here. Kind of the anatomy of a virus. But the most important thing is that they can't replicate on their own. They are non-replicating compounds. And so what they do is they infect a host, so like an animal or a plant or something. They infect them by injecting their DNA or their RNA in them, and using the cellular machinery, the enzymes, the ribosomes, things like that, of the host cell to basically replicate the DNA or the RNA of the viruses, and to transcribe and translate those into proteins and make more viruses. And so, like I said, viruses have a simple construction. There's many different types, but they have some sort of protein covering. It's called a capsid or a capsomere. So in this helical virus, it's the spiral protein coating that coats the inside. This is the capsid. This kind of polyhedral virus, this is the capsid right here. And in this is a complex virus, or kind of one that would be a phage. Capsid is up here. It kind of protects the genetic material. Now inside the capsid is basically the genetic material. It can be DNA or it can be RNA. There are DNA and RNA viruses. And you can see that we've got several things going on here in the phage. You got a sheath and some pins and tail fibers. This is a very complex virus, but mostly to be a virus you just need a capsid that protects the DNA or the RNA. Because they're basically just protein coated DNA or RNA, and they can't self-replicate, most scientists agree that viruses aren't alive. They're not self-replicating, they're not alive. It's a big kind of philosophical point of contention in virology, is whether they are alive or not, but the general consensus is they're not. So this is just some general anatomy of a virus. Viruses also can have these surface receptors, glycoproteins on the outside, and you probably actually heard of them, strangely enough. A lot of viruses, like the flu virus, have 2 different kinds of surface receptors they use to glom on to different cells and infect them. And they're called neuraminidase and hemagglutinin. And this is where the N and the H come from in like flus like H1N1. So like H1N1, that's just saying 1 is a number to specify which of a number of different hemagglutinin receptors that has, so an H1N1, it has hemagglutinin type 1 receptors, and it has type 1 neuraminidase receptors. So H6N1 has type 6 hemagglutinin receptors and type 1 neuraminidase receptors. So they're just a type of surface receptors that certain flu viruses have. Just thought that might be really interesting for you to know now when they talk about it in the news or whatever. So the life cycle of a virus, basically it's really simple what they do to keep replicating themselves. First, this virus needs to gain access into the cell. So they do that by either injecting their DNA or RNA through the cell into the cytoplasm, or by kind of merging their capsid with the plasma membrane and injecting the DNA and the capsid proteins in there. Once the DNA is injected into, or the RNA is injected into the cell, it uses the cell's enzymes. So the ribosomes, the polymerases, the DNA polymerases, the ligases, all of that to replicate its DNA. So it's replicating its DNA. And it's transcribing some of that DNA into mRNA, which makes more capsid proteins. And then inside the cell, the replicated DNA and those capsid proteins reassemble into small little viruses that then break out of the cell and can go in and infect new host cells. So that's really, in a nutshell, the life cycle of a virus. Infects, replicates its DNA, and translates and transcribes mRNA into new viruses that can go out and infect new cells. So that's the basic life cycle. Now we can break viruses up into two smaller groups by there's slight variation on that life cycle. They're called lytic and lysogenic bacteria, or lysogenic phases. So there's two phases or possible life cycle patterns in a virus, the lytic and the lysogenic phase. In one phase, and I would say phases because these two life cycles can happen with the same virus in turn. They can butt off of each other, kind of like this figure 8 goes. So we start by, the virus injects its genetic material. And what happens after that is the genetic DNA circularizes or forms a circle. It kind of matches the bacterial plasmoid or a plasmid. And at this point, there's a fork in the road. Certain environmental and internal factors can induce this host bacteria and the viral DNA within it, or genetic material within it, to go in two different paths. In one path, it can go into what's called the lytic cycle, which is basically what we talked about in this previous slide. The DNA or RNA gets translated, transcribed into proteins, gets replicated, makes new viruses, and those viruses multiply until they burst or lyse the cell, destroy it, and go on and to infect many other cells. If those certain factors aren't present, or different factors are present which push the viral genetic material to a lysogenic cycle, and this is basically a dormant cycle. So what happens is this viral DNA integrates itself into the bacterial plasmoid or chromosome, and it becomes what's called a prophage. So that bacterial genetic material is bound on, or it becomes part of the bacterial genetic material. And it lays there and it's dormant. And the bacteria goes and replicates through binary fission, makes more and more bacterial new daughter cells, each of which have a copy of the viral DNA just sitting dormant in their cell. Then at some point, occasionally one or more or all of these prophages with the viral DNA embedded in there receives a signal and can spontaneously jump in and initiate the lytic phase. And all of these cells can be destroyed and produce viruses at once. Pretty scary. You see this in a lot of serious viral infections where they might get infected and then years later, boom, you have a serious illness, a serious symptoms because some sort of environmental or internal signal has told all of these infected cells, these prophages, to spontaneously initiate the lytic or induce the lytic cycle of their lifetime. So lytic, lysogenic can go hand in hand. Some bacteria only are lytic, some are lytic and lysogenic like this. The last thing virally I want to talk about is the case of HIV. Now, HIV is a very well-known and serious virus, past sexually transmitted virus. And one of the things that make it so difficult to combat is that it's hard to kill all the virus. It's able to replicate even after the virus that's initially infected you has been destroyed. Now, one of the reasons is because it's an RNA virus. HIV virus, or the HIV, contains RNA instead of DNA. But most importantly is it carries an enzyme called reverse transcriptase. And what this enzyme does is it changes the viral RNA, once it's been injected in the host cell, into DNA. So transcription goes from DNA to RNA, reverse transcriptase goes from RNA to DNA. And that DNA becomes combined with the host DNA. And then every time the host cell goes through mitosis, it creates more and more cells that have that viral DNA in it that can eventually be turned back into mRNA, transcribed and translated into other viruses that can continually infect you. So here in this picture, we have this virus injecting its RNA and its reverse transcriptase enzymes into the host cell. Then the reverse transcriptase starts reverse transcribing that RNA into host double-stranded DNA, which then becomes a provirus. It becomes part of the host DNA. And then it can be transcribed as many times as it wants into new DNA cells. This is why, or one of the many reasons why HIV is so hard to cure, so hard to find treatments for. Almost all of the current treatments we have for HIV are reverse transcriptase inhibitors, which means they make this enzyme reverse transcriptase not work. And then we can battle it more like a regular virus that doesn't contain reverse transcriptase. Last thing I want to talk about is genetic variation in bacteria. So you might be wondering, well, bacteria reproduce asexually through binary fission. So there's no crossing over, there's no recombination of genetic material. There's no sexual recombination, getting some genetic material from mother and father. So how can bacteria evolve? How can they acquire new traits? How can they survive? And while it's true mutations can happen, and they actually happen faster in some viruses because the same proofreading mechanism for genetic material isn't present in bacteria. And bacteria with RNA as their primary genetic material seem to show a lot higher rates of genetic mutation. But the main way is that they can exchange genetic material laterally, which means between each other in the same generation. So instead of vertical genetic exchange, like from parent to offspring, they can exchange genetic variation between each other. Like if I could exchange genetic material with my own siblings. What this allows bacteria to do is evolve very quickly, because any trait that or allele that is beneficial can be exchanged with members of the same generation. And this can happen in 3 different ways. One is called bacterial transformation. Genetic material, especially on the plasmid or small circular parts of the DNA, can directly go through transformation outside of the bacterial cell and be absorbed into a new cell, a new bacterial cell, and incorporated and replicated. So it can be like passed from bacteria to bacteria through interstitial fluid, through empty space. And we'll be doing this in the lab, so we'll be doing bacterial transformation in the lab. Another way genetic material can be exchanged between bacteria is what's called bacterial transduction. There are special viruses that mainly or only infect bacteria called bacteriophages. And what they can do is they can inject their genetic material into a bacteria and replicate all the DNA, and then the bacteriophage that's created can have some bacterial DNA from this bacteria in it, and when it infects a different recipient bacteria, inject some of that uncommon DNA that was part of this bacteria cell, but not this one, into this new bacterial cell, and create a new recombinant bacterium. In the last part, or last way bacteria can share genetic material, this is how antibiotic resistance by and large is shared. It's called conjugation. So the bacteria cells can kind of merge their cell walls like this, and a whole plasmid can be shared in between. This whole circular plasmid and all of its DNA can go over to this new recipient cell, and it can be replicated and produce those same genetic material, those same genes in this new bacteria cell. So even though bacteria cannot sexually reproduce, they have various ways that they can exchange genetic material with each other.